Yes, today we are going to see another 5 mark short notes for either paper 1 or paper 2, because this is coming under either plasma protein metabolism question, or sometimes it is coming under instrumentation like electrophoresis, right? So this electrophoresis, what are all the subtitles? First you should know about the subtitles for this, because it is your 5 mark question, a definite 5 mark question, no essay question. So you should write minimum points for the electrophoresis, because it is actually PG question, MD Biochemistry PG question, but you should know the basics or outline of electrophoresis. Okay, so before going to the electrophoresis, name the plasma proteins. It is very important Viva question and also MCQ question. The plasma proteins are albumin, globulin and fibrinogen, right? We are having some normal levels for all these, that is total protein 6 to 8 gram per DL. Here albumin is 3.5 to 5.5 grams per DL, whereas globulin 2.5 to 3.5 grams per DL. If you are adding these two, definitely total protein will come. You should know the difference. Sometimes some students what you are doing is total protein is some 10, but addition of this globulin and albumin is less than 5 or 6, like that only, right? If you are adding these two means that this will come. So total protein is 6 to 8, 3.5 to 5.5 grams per deciliter for albumin. Some books say 6 also, right? Globulin 2.5 to 3.5. Fibrinogen, it is different, the unit is milligram per deciliter, 200 to 400 only. All the 4 values are very important in MCQs, right? Fibrinogen 200 to 400 milligram per deciliter. I am giving some examples for all these globulin fractions. In globulin we are having 4 fractions, that is alpha 1, alpha 2, beta and gamma. Under gamma, the immunoglobulins are there. Under beta globulin, C reactive protein and beta 2 microglobulin, whereas alpha 2 ceruloplasmin and alpha 2 macroglobulin, whereas alpha 1 is alpha protein and alpha 1 antitrypsin. So these are all some examples. So next one is, with this albumin globulin we are having one ratio, that is synthesis of albumin and globulin. Usually albumin is little bit of high, globulin is some low. Globulin is fixed, only 1, whereas albumin is, it starts from 1.2 and ends with 1.6 only, that is 1.2 to 1.6 is the albumin level, whereas globulin level is 1. Why you want to measure this AG ratio means, in some diseases or some disorders, the AG ratio will be reversed. That is a clinical important point, okay? Usually the AG ratio is 1.2 albumin or sometimes 1.6 albumin, but globulin is always 1. In some clinical diseases or conditions, the ratio will be reversed. That is, albumin level is low, globulin is increased, right? Now, what is the picture of electrophoresis? So this is the picture. Either this will be presenting on electrophoresis paper, that is Wattmann's filter paper, or sometimes agar gel. This is the picture of electrophoresis in a normal person, right? So this is the point of application. This is the negative side, this is the positive side. You are attaching one electric apparatus with the power supply. You are attaching the 2 ends in the 2 plug point side. One is negative charge, another one is positive charge. Here this is a place for point of application. That means you have to apply the patient's serum or patient sample in this area. After switch on the apparatus, the particle, that is protein or any amino acid or immunoglobulin or lipoprotein, they will be moving from the negative to positive side, depending upon their size, shape, molecular weight, like that. Okay. After 1 hour, after 2 hours, after 48 hours, like that. There we have so many procedural techniques. But in this electrophoresis, this is 1.5 hours for the page electrophoresis. Okay. So the time is also nowadays high voltage electrophoresis also come. The time is a very minimal time only. So this slide shows the electrophoresis with the normal patient. They call it as band, albumin band, globulin band, like that only. Okay. These are all the bands. First one is point of application here is gamma globulin, then beta, then alpha 2, alpha 1 and albumin. Why albumin is in the last means albumin is having very minimal molecular weight, that is why it is moving slowly to the last end of the slide. Otherwise gamma globulin is more molecular weight, so that will very closely settled in the negative side. This is a slide shows the normal serum electrophoresis. Okay. So what are all the subtitles for this electrophoresis question? First one is you have to define or give some introduction about the electrophoresis. Second one is who invented this technique and which year you have to mention, just one line. Then what is the principle? For any technique in biochemistry, you have to mention 2 or 3 lines about the principle. What happens really in that electrophoretic machine? What is the mechanism behind the machine? Like that you have to give 2 or 3 lines for principle. Next one is what is the procedure? It will take 1.5 hours, means every minute, every 5 minutes, every hour, what will happen in that movement of the particles you have to mention. But for you just give the outline only, don't worry about the procedure because it is for PhD students or MD students only. So don't worry about or don't get confused while reading procedure of electrophoresis, right? I am also going to give some outlines only about the electrophoresis. That is enough for you. Next one is this procedure, actually some factors affect this procedure. So what are all the factors? Then finally you will get a result. Usually the result will be read by densitometer. In the biochemistry lab they are having densitometer. After completing this procedure, you have to apply the slide or paper in that densitometer. Finally you will get the band size, band color, band width, like that you can measure. Next one is interpretation. Interpretation means what is a normal slide, what is the abnormal slide. If you have an abnormal slide, means what are all the diseases involved in this abnormal slide. Kidney condition, liver condition, bone condition or infections, like that you have to interpret the slide with available bands. Suppose albumin band is very minimal, not like in a normal serum electrophoresis, means the patient is having low albumin. So the problem is in the liver because albumin is synthesized in the liver. So for any diseases in liver means albumin level definitely will be low. So first point is liver diseases. For example, cirrhosis of liver. Second point is suppose the globulin is high, the microglobulin is available, means the problem is in kidney. That is nephrotic syndrome. Like that multiple myeloma, some M band, they will be call it as M band, which is the marker or identity of multiple myeloma. Like that you have to interpret the available result of electrophoresis. And next one is types. What are all the types available in electrophoresis? And last one is applications. Because why I am underlining this means sometimes they will be asking applications of electrophoresis only. Very simple, very easy. Right, 4 or 5 applications I am giving. That is enough for your 5 mark question. Okay. Yes, the introduction for electrophoresis is the movement of charged particles. Particles they are having either low charge or high charge, like that. So movement of charged particles, charge may be whatever, particles when subjected to an electric field, it is passing through the current passing through the protein or charged particles. Okay, positive ions move towards cathodes, you know the mechanism, whereas negative ions always move towards anodes. So we have two, it's a cathode and anode. Positive ion goes to this side and negative ion goes to this side. So movement of charged particles when subjected to an electric field, they will move on, that is positive ions move towards cathode whereas negative ions move towards anode. That is the compounds in the mixture, you are applying one mixture of compounds for the separation of proteins. In that mixture, they move in relation to their net charge psi. size, shape, mass, molecular weight, and then get separated. There are so many factors for the movement and also separation of these particles in your desired mixture, so that the desired compound can be identified and separated or isolated. Okay, so this electrophoresis techniques was invented by Tiselius in 1933 for the separation of plasma proteins. Okay, yes, now we are going to see what are all the factors which will affect the electrophoresis procedure. Okay, so the first one is size and shape of the particle. If the particle size is small, means the movement will be rapid, whereas the size of the particle means huge, movement will be low. That is why size and shape of the desired that is measured particle. Second one is temperature. Temperature is very, very important. It will also affect the movement of the particle. Third is net charge of the particles. It will also affect the electrophoretic procedure. Next one is electric field is very, very important. That is 180 to 200 volts of temperature is very, very important. If you have any situation like breakdown of or power cut situation, definitely the electrophoretic procedure will get affected. The last one is properties of the supporting media. We are having so many supporting medias like paper electrophoresis, agar electrophoresis, gel electrophoresis, like that. If there is any problem with that property or quality of the supporting media, means this will also affect the electrophoretic procedure, right? So these are all the factors affecting electrophoresis procedure. Next one is what are all the types? We have 4 or 5 types. You just mention the name of the types. Don't worry about the procedure and principle for others. Okay, because they ask mention 2 types of other electrophoretic types, or mention any other type of like that only they will be asking for 1 mark or 2 mark question only. For that question you have to mention the type 1 is paper electrophoresis, type 2 is page, that is polyacrylamide gel electrophoresis. Next one is isoelectric focusing, and then capillary electrophoresis, finally immunoelectrophoresis. You should know these 5, at least you should know about these 5 names, because they are asking 2 or 3 for the type of electrophoresis question. Okay, in your clinical scenario or sometimes theory point also it will be useful for you. Then coming to the applications where we are using this electrophoretic procedure, we know the first one is separation of plasma proteins, right? Like that we are having lipoprotein, isoenzyme, immunoglobulins, nucleic acid, DNA and RNA, and some abnormal hemoglobin like sickle cell anemia. So these are all the applications for the electrophoretic procedure. Okay. Second one is to find out the size of the protein, and quantification of the protein also measured by this electrophoretic technique, and to identify the sulfide bonds, whether it is minimal or whether it is abnormal, like that we can identify the sulfide bonds present in the polypeptide chain or any structure of the protein, and then to get the purity of the samples. So these are all the applications for electrophoresis. You at least try to give 3 or 4 applications for electrophoresis. Okay. Yes, next we are moving on to supporting media. Supporting media means it is nothing but where you are going to apply your sample, that is patient's serum, and where the electrophoretic procedure will take place, that is supporting media. We are having 4 supporting media. You just know the name of 1 or 2 because you have to write it in your procedure for your electrophoresis. So first one is paper, that is Wattsman filter paper. The name of the paper is Wattsman filter paper. Second one is agar. It only produces gel. Over the gel only you are going to apply the patient sample. Third one is cellulose acetate. And finally page, polyacrylamide gel electrophoresis. These 4 are coming under the supporting media for your electrophoresis. Next one is How will you interpret it? Before going to the interpretation, what is the procedure? I am just giving the outline only. You just go through 1 or 2 times, definitely it will be easy for you. First you take one electrophoretic apparatus which is having one buffer tank, that is 2 troughs. 2 sides, 2 troughs, that is totti. Mean totti, in the totti Muralis. So 2 troughs. Then you have to add barbitone buffer in the 2 troughs at the pH of 8.6. For electrophoresis, the pH is uniform, 8.6 only for barbitone buffer. Next one is you should have power supply of 180 to 200 volts, continuous power supply, right? No power break or power cut. Otherwise it will get disturbed. Next one is you have to take either Wattman filter paper or agar gel. You have to prepare the gel from the agar. Then apply the blood sample in the point of application area. Then you are going to use one dye, because for the coloring reagent only you have to monitor the procedure after 1 hour, after 2 hours, whether it is moving or not, whether it is obstructed or blocked, like that you have to find out. The procedure means you have to apply one dye, bromophenol blue. Then addition of staining and destaining. After procedure is over, you have to apply some staining agents. Then you have to put it in the hot air oven for the 110 degree temperature, like that. Then you have to take out, add staining and destaining agent, like that. Finally, you have to apply one fixative solution for the band fixation. After this fixation only, the band will get fixed in separate areas according to their size, mass, molecular weight, like that. Finally, you will get the picture that is called electrophotogram. In the paper electrophoresis, you can read the diagram, you can read the bands. Whereas in page and agar gel electrophoresis, you should apply this electrophoretic slide into the densitometer. That machine will give the density of the band, color of the band, width of the band, like that. It will give the printout. Right? That is why densitometer is, nowadays we are using densitometer only for the study of the bands of electrophoresis. Okay, after getting this electrophoretic result, it looks like already first slide I have seen one normal serum sample electrophoresis, no, albumin, globulin, like that. So now you are having the slide, how you are going to interpret it, whether the slide is normal or the patient is having some abnormal, like that, right? We are going to have only 4 abnormal conditions, right? Usually the first one is chronic infections. Any infection, the liver albumin synthesis will be low. For compensatory mechanism, globulin will be raised. First condition is chronic infections. Second one is due to alcoholism, most of the patients having cirrhosis of liver. For that patients, clinicians always advise or want to go for electrophoresis. In that time, the patient's slide shows decreased albumin because of the albumin liver problem. Because of the cirrhosis, the albumin synthesis will be low. So definitely in the electrophoretic field also, band or width of the albumin is very low. Compensating mechanism, gamma globulin is increased. This is for cirrhosis of liver. Third one is nephrotic syndrome. Here we are having some problem in the glomerular basement membrane, problem in the filtration of the alpha 2 macroglobulin, like that. So albumin low, alpha 2 globulin high. Albumin low, alpha 2 globulin high. Why? So the alpha 2 macroglobulin molecular weight is so high, it is not able to filter or through excretion through kidney. That is why albumin is less, whereas alpha 2 globulin is high. This is the only marker or only indicator for nephrotic syndrome in electrophoresis. And in the special condition, one is our bone condition, osteolytic condition, multiple myeloma. Here what happens is special band, M band, multiple myeloma, MM. So band name is also M, multiple myeloma, M band, which is present in between the beta and gamma region, right? So these 4 are the abnormal conditions. You should interpret, you should find out whether your patient is normal or abnormal, right? First you have to see the albumin picture. Width and band of albumin shows whether the albumin synthesis is less or normal. If it is normal means go and look for alpha 1 and alpha 2. Alpha 2 is the routinely or commonly occurring situation nephrotic syndrome. The band will be raised or increased. Like that you have to interpret all the 4 conditions for electrophoresis. Okay.